How are tree rings used to date historical events?
The study of tree rings is known as dendrochronology. Every year, trees produce an annular ring composed of one wide, light ring and one narrow, dark ring. During spring and early summer, tree stem cells grow rapidly and larger, thus producing the wide, light ring. In winter, growth is greatly reduced and cells are much smaller, producing the narrow, dark ring. In the coldest part of winter or the dry heat of summer, no cells are produced. Comparing pieces of dead trees of unknown age with the rings of living trees allows scientists to establish the date when the fragment was part of a living tree. This technique has been used to date the ancient pueblos throughout the southwestern United States. A subfield of dendrochronology is dendroclimatology. Scientists study the tree rings of very old trees to determine climatic conditions of the past, the effects of droughts, pollution, insect infestations, fires, volcanoes, and earthquakes are all visible in tree rings.
What is the founder effect?
When a small group of individuals forms the genetic basis for a new population, only the variations within that founding group will be found in the resultant population. This is known as the founder effect. An example of the founder effect is found among the finches of the Galapagos Islands.
What are sex chromosomes?
Sex, X and Y in mammals, chromosomes are the non-matching pair. The Y chromosome, found only in males, has the sex-determining gene, SRY. The SRY determines whether or not an individual will develop testes and produce appreciable quantities of testosterone, the hormone that generates male characteristics. There is no known correlate gene on the X chromosome for the formation of ovaries. Therefore, it is the absence of the Y chromosome that determines a female.
Do different fungi rot different parts of trees?
Most fungi that rot wood in standing trees are basidiomycetes. The group that includes mushrooms, fleshy shelves, and rigid brackets. Most wood rotters attack only one or two related species of trees. Conifers and deciduous trees are more prone to fungi damage. Many wood rot fungi damage only specific parts of a tree. For example, Ganoderma lucidum and Heterobasidion annosum specifically rot roots and are rarely found in higher sections of trees. Sirena unicolor and Climacodon septentrionale are common in high sections of trees and are rarely found in the roots. Laetoporus sulphurus and Fomitopsis pinicola are found in all sections of a tree except the smallest branches of their host.
Can an innocent person be convicted based on DNA analysis?
Current methods of DNA analysis are very sensitive, as only a few cells are needed for DNA extraction. However, it is possible for an innocent person's DNA to be found at any crime scene, either from accidental deposition or by direct deposition by a third party. Also, a partial DNA profile from a crime scene could match that of an innocent person whose DNA is already in a DNA data bank. In addition, close relatives of suspected criminals could also be partial matches to a DNA profile.
What is adaptive radiation?
As populations move into new environments and adapt to those local conditions, there is an increase in diversity. This splitting creates a divergence from the original population. When diagrammed on paper, the new populations appear to be radiating outward from the original like the spokes of a wheel. An iguana in the Galapagos Islands. Because island populations tend to be reproductively isolated from the mainland, they are more likely to demonstrate adaptations that are particular to that island's ecology.
Who first proposed that the cell membrane is composed of a bilayer structure of lipids?
In 1925 Evert Gorter, 1881–1954, and F. Grendel, two Dutch physiologists, hypothesized that there is a bilayer structure of lipids on the cell surface. Their work was significant because it was the first attempt to understand membranes at the molecular level.
When were chloroplasts first described?
Because chloroplasts are a large cell structure, bigger than any other organelle except the nucleus, they were described and studied early in the history of cytology. Anton van Leeuwenhoek, 1632–1723, and Nehemiah Grew, 1641–1712, described these organelles in the 17th century. In the early stages of the microscopic study of cells,
What is an ecological pyramid?
if the organisms in a food chain are arranged according to trophic levels, they form a pyramid, with a broad base representing the primary producers, and usually only a few individuals in the highest part of the pyramid. Also known as a pyramid of numbers, An ecological pyramid is a way of describing the distribution of energy. Biomass, or individuals among the different levels of ecosystem structure.
How has centrifugation been used in biological applications?
Biologists primarily use centrifugation to isolate and determine the biological properties and functions of subcellular organelles and large molecules. They study the effects of centrifugal forces on cells, developing embryos, and protozoa. These techniques have allowed scientists to determine certain properties about cells, including surface tension, relative viscosity of the cytoplasm, and the spatial and functional interrelationship of cell organelles when redistributed in intact cells.
Did Darwin and Mendel know each other?
Although both men lived during the same time period in the 19th century, they did not know each other. Charles Darwin's, 1809–1882, publication on the origin of 387 species, 1859, popularized his theory of natural selection but raised many questions as to how organisms could display modified or new traits. In 1865 Mendel published his landmark paper Experiments in Plant Hybridization. Researchers have been unable to document whether either man used the other's work in the development of their respective theories.
What is the Hardy-Weinberg theorem?
The Hardy-Weinberg theorem, formulated in 1909, extends Mendel's laws to natural 392 populations. It explains in a formula what happens to the allelic and genotypic frequencies as alleles are passed between generations. The Hardy-Weinberg equilibrium is based on the following assumptions: 1. The population size is large. 2. Individuals mate randomly within the population. 3. There are no mutations. 4. There is no input of new alleles from outside sources, and 5. There is no natural selection, such that each new allelic combination has equal chances of survival.
How many nuclear pores are there in a nucleus?
A typical nucleus in a mammalian cell has 3,000 to 4,000 pores or 10 to 20 pores per square micrometer, m. Nuclear pores are not merely perforations, but channels composed of more than 100 different proteins.
What is the struggle for existence?
As noted by Thomas Malthus, 1766–1834. More individuals are born than can possibly survive due to limited resources. Therefore, there is a struggle for existence. We can think of this as the competition between individuals, of the same species or between species, for food or hiding places or other necessary resources. The origin of this phrase can be traced back to the Greek philosophers but is perhaps best described by Alfred Lord Tennyson's, 1809–1892, phrase, "Nature red in tooth and claw."
What is ADP?
ADP, adenosine triphosphate, is the universal energy currency of a cell. Its secret lies in its structure. ADP contains 3 negatively charged phosphate groups. When the bond between the outermost 2 phosphate groups is broken, ADP becomes ADP, adenosine diphosphate. This reaction releases 7.3 kcal/mol of ADP, which is a great deal of energy by cell standards.
What are some biological applications of chromatography?
Chromatography has many applications in biology. It is used to separate and identify amino acids, carbohydrates, fatty acids, and other natural substances. Environmental testing laboratories use chromatography to identify trace quantities of contaminants such as PCBs in waste oil and pesticides such as DDT in groundwater. It is also used to test drinking water and test air quality. Pharmaceutical companies use chromatography to prepare quantities of extremely pure materials. The food industry uses chromatography to detect contaminants such as aflatoxin.
What plants produce essential oils that are commonly used in aromatherapy?
Aromatherapy is a holistic approach to healing using essential oils extracted from plants. Holistic medicine looks at the health of the whole individual, and treatments emphasize the connection of mind, body, and spirit. The term aromatherapy was first used by René Gattefossé, a French perfume chemist. He discovered the healing powers of lavender oil following a laboratory accident during which he burned his hand. Gattefossé began to investigate the properties of lavender oil and other essential oils and published a book on plant extracts. During aromatherapy treatments, essential oils are absorbed through breath or the pores of the skin. This process triggers certain physiological responses.
What is a hybrid?
A hybrid is produced as the offspring of 2 true breeding organisms of different strains, AAXA. If the hybrids, AA, are then mated, the result is a hybrid cross.
Why are arthropods considered the most biologically successful phylum of animals?
Members of the phylum Arthropoda are characterized by jointed appendages and an exoskeleton of chitin. There are more than 1 million species of arthropods currently known to science. And many biologists believe there are millions more to be identified. Arthropods are the most biologically successful group of animals because they are the most diverse and live in a greater range of habitats than do the members of any other phylum of animals.
What does species diversity have to do with evolution?
Species diversity is direct evidence that evolution has occurred. When species can be identified in which individuals share a number of significant traits, while also having some unique adaptations, it is logical to assume that the common traits are the result of common origin while the unique ones demonstrate adaptive radiation. An example of the significance of species diversity would be Darwin's finches of the Galapagos Islands. While the species share a common body structure that is inherited from a common ancestor, each species also demonstrates variations in beak size and structure that are indicative of their adaptations to local environments and the type of food available.
Who proposed that Earth was only 6,000 years old?
In the 17th century, Archbishop James Ussher, 1581–1656, of the Irish Protestant Church, calculated from the Old Testament genealogies that Earth was created on Sunday, October 26, 4004 CE. He was also able to calculate exact dates for when Adam and Eve were forced from paradise, Monday, November 10, 4004 BCE, and when Noah's Ark touched down on Mt Ararat, Wednesday, May 5, 1491 BCE.
What is meant by genetic discrimination?
Genetic discrimination refers to the use of genetic information by health insurers and employers to determine eligibility, set premiums for health insurance, or hire and fire people. The U.S. Senate passed the Genetic Information Non-Discrimination Act of 2003 on October 14, 2003. The Act establishes basic legal protections that will prohibit discrimination in health insurance and employment on the basis of predictive genetic information. It will fully protect the privacy of genetic information.
What is the RNA Tie Club?
In 1953, shortly after the publication of James Watson and Francis Crick's paper on the structure of DNA, George Gamow, 1904–1968, a Russian physicist, wrote to Watson and Crick and suggested a mathematical link between DNA structure and the structure of 20 amino acids. The RNA Tie Club was an outgrowth of this proposal. Members included Watson, Crick, and 17 others, each taking a nickname from one of the 20 amino acids. Each member of the club was presented with a specially designed tie corresponding to the appropriate amino acid.
How are generations described in genetic crosses?
Usually, genetic crosses begin with true breeding parents of different strains that are designated by letter P. The hybrid offspring are called the F1 generation, each subsequent generation is called F2, F3, and so on.
Why was Stewart successful?
Like previous investigators, he supplied the cultured cells with sugars, minerals, and vitamins. In addition, he also added a new ingredient, coconut milk. Coconut milk contains, among other things, a substance that induces cell division. Subsequent research identified this material as cytokinins, a group of plant hormones, growth regulators, that stimulate cell division. Once the cultured cells began dividing, they were transplanted on agar media, where they formed roots and shoots and developed into plants.
Which lichens are most often used by the perfume and cosmetic industries?
The musk-like fragrance and fixative properties of Avernia prunistri and Pseudavernia furfurnacea make them popular components for perfumes and cosmetics. The essential oils of these lichens are extracted with solvents E. prunistri and P. furfurnacea are common to southern France, Morocco, and the Serbo-Croatian peninsula.
How does a primary cell wall differ from a secondary cell wall?
A primary cell wall is laid down during cell division and is relatively thin and flexible in order to accommodate cell enlargement and elongation. It is strengthened when the cell matures and stops growing. A secondary cell wall is present between the plasma membrane and primary cell wall in some cells. The secondary wall is often deposited in several laminated layers. It is strong and durable, and provides both cell protection and support. Wood consists mainly of secondary cell walls.
What are the functions of the cell membranes?
Cell membranes define and compartmentalize space, regulate the flow of materials. detect external signals, and mediate interactions between cells.
What technique may be used to determine the presence and concentration of various chemicals in a solution?
Spectrophotometry is based on the principle that every different atom, molecule, or chemical bond absorbs a unique pattern of wavelengths of light. Using a spectrophotometer, scientists are able to measure the amount of light that is absorbed or transmitted by molecules in solution. This value of this measurement allows scientists to identify the chemical.
What is a double-blind study?
In a double-blind study, neither the subjects of the experiment nor the persons administering the experiment know the critical aspects of the experiment. This method is used to guard against both experimenter bias and placebo effects.
How does one prepare a 110 dilution?
A 110 dilution means 1 part in a total of 10. There are 3 different ways to prepare a 110 dilution. 1. The weight-to-weight, WW method. 2. The weight-to-volume, WV method, and 3. The volume-to-volume, VV method. In the weight-to-weight method, 1.0 g of solute is dissolved in 9.0 g of solvent, yielding a total of 10 parts by weight, 1 of which is solute. In the weight-to-volume method, enough solvent is added to 1.0 g of solute to make a total volume of 10 ml. In this method, 1 part, by weight, is dispersed in 10 total parts, by volume. Since most biological solutions are very dilute, the accuracy of most research is not affected if a previously weighed solute is dissolved in the desired volume of solvent. The volume-to-volume method is preferred when the solute is a liquid. 1 milliliter of solute, such as ethanol, added to 9.0 ml of water yields a 10-part solution, 1 part of which is the solute.
How many Nobel Prizes have been awarded in genetics?
Of the 162 Nobel Prizes awarded in the category for physiology or medicine, 42 prizes, 26%, have been awarded in genetics. This far exceeds other categories. The closest in number is disease treatment, with 24 prizes, 15%.
How were fungi involved in World War I?
During World War I, the Germans needed glycerol to make nitroglycerin, which is used in the production of explosives such as dynamite. Before the war, the Germans had imported their glycerol. But the British naval blockade during the war prevented such imports. The German scientist Karl Neuberg, 1877–1956, knew that trace levels of glycerol are produced when Saccharomyces cerevisiae is used during the alcoholic fermentation of sugar. He sought and developed a modified fermentation process in which the yeast would produce significant quantities of glycerol and less ethanol. The production of glycerol was improved by adding 3.5% sodium sulfite at a pH of 7.0 to the fermentation process, which blocked one chemical reaction in the metabolic pathway. Neuberg's procedure was implemented with the conversion of German beer breweries to glycerol plants. The plants produced 1,000 tons of glycerol per month. After the war ended, the production of glycerol was not in demand, so it was suspended.
What are the agricultural uses of antibiotics?
Antibiotics are sprayed onto fruit trees and other food-bearing plants to control disease. In addition, antibiotics are added to feedstocks to prevent disease and improve the growth rate among food-producing animals.
What is the most abundant group of organisms?
The eubacteria are the most abundant group of organisms on Earth. More living eubacteria inhabit the human mouth than the total number of mammals living on Earth.
Who extensively studied fly behavior?
Vincent de Thier, 1914–1993, spent most of his life researching chemical responses in insects. He wrote numerous articles and books, both for the general public and specifically for children. To Know a Fly, perhaps his most famous book, is considered one of the classic books in entomology. The book details physiological research on flies, particularly with regard to chemoreception, with a touch of humor. Aside from his career as a distinguished research scientist, he was able to popularize what scientists do and what draws people to science.
Which organelle is absent in fungi?
Centrioles, which divide and organize the spindle filters during mitosis and meiosis, are lacking in all fungi. The nuclear envelope does not break down and reform. Instead, a spindle apparatus is formed within the envelope. Fungi regulate the formation of microtubules during mitosis with small amorphous structures called spindle plagues.
How do skin cells keep your blood from leaking out?
The skin is comprised of multiple layers of cells. The outermost layers are made of dead cells full of keratin. Sebaceous glands coat these dead cells with an oily secretion that makes them water resistant. However, they are not waterproof. About 1 pint of fluid from deeper tissues leaks through the skin's surface every day and evaporates. This excretion is in addition to perspiration produced by excessive heat or strenuous activity. The very strong junctions holding the cells together, known as desmosomes, prevent large amounts of fluid leakage across the skin barrier. The linkages are so efficient that the epidermal skin cells tend to slough off in sheets rather than individually.
What is the role of the queen bee?
A honeybee society is made up of a single queen, a few male drones, and up to 80,000 female workers. The queen is the only female in the hive capable of reproduction, and she can lay as many as 1,000 eggs in a single day. While the queen's job is solely to lay eggs, the workers' jobs change over time. Young workers serve as nurses to the larvae that develop from the eggs. Older workers serve first as hive builders and then as foragers.
What type of cell is found in nerve tissue?
Neurons are specialized cells that produce and conduct impulses, or nerve signals. Neurons consist of a cell body containing a nucleus and two types of cytoplasmic extensions, dendrites and axons. Dendrites are thin, highly branched extensions that receive signals. Axons are tubular extensions that transmit nerve impulses away from the cell body, often to another neuron. Other cells in nerve tissue nourish the neurons, insulate the dendrites and axons, and promote quicker transmission of signals.
What invertebrate lives in both marine and fresh waters and is one of the most important of all animals?
Copepods, tiny crustaceans, are the link between the photosynthetic life in the ocean or pond and the rest of the aquatic food web. They are primary consumers grazing on algae in the waters of the oceans and ponds. These organisms, among the most abundant multicellular animals on Earth, are then consumed by a variety of small predators, which are eaten by larger predators, and so on. Virtually all animal life in the ocean depends on the copepods, either directly or indirectly. Although humans do not eat copepods directly, our sources of food from the ocean would disappear without the copepods.
Do animals laugh?
So far, No example of animal laughter comparable to that of humans has been documented. However, researchers have reported that under certain conditions some species emit special vocalizations that could be considered akin to laughter. In one specific case, a researcher has been able to identify a huffing sound unique to dogs at play. Scientists studying the behavior of non-human primates and rats have reported similar observations.
What are the main components of the cell wall in plants?
The cell wall is one of the features of plant cells that distinguishes them from animal cells. The wall protects the plant cell and maintains its shape. The cell wall consists mainly of cellulose microfibrils embedded in a matrix of protein and sugar polymers.
What groups of organisms have a cell wall?
A cell wall is present in organisms in the kingdoms Archaebacteria, Eubacteria, Protista, fungi, and Plantae. Animals are the only organisms that do not have a cell wall.
How do fungi reproduce?
Fungal reproduction occurs in two different ways: sexually or asexually. Asexual reproduction occurs through fission, budding, or most commonly by spore formation. Sexual reproduction occurs by means that are characteristic for each group. Two types of reproductive structures are found in fungi. Sporangia produce spores, whereas gametangia produce gametes. In order to sexually reproduce, fungi often carry out some type of conjugation. Hyphae of two genetically different mating types come together and fuse, forming a diploid zygote. Most fungi reproduce sexually with nuclear exchange rather than gametes.
Who proposed using magnetic resonance imaging for diagnostic purposes?
The concept of using MRI to detect tumors in patients was proposed by Raymond Domitian. 1936, in a 1972 patent application. The fundamental MRI imaging concept used in all present-day MRI instruments was proposed by Paul Lauterbur. 1929, in an article in Nature in 1973. Lauterbur and Peter Mansfield, 1933. Were awarded the Nobel Prize in Physiology or Medicine in 2003 for their discoveries concerning magnetic resonance imaging. The main advantages of MRI are that it not only gives superior images of soft tissues, like organs, but it can also measure dynamic physiological changes in a non-invasive manner, without penetrating the body in any way. A disadvantage of MRI is that it cannot be used for every patient. For example, patients with implants, pacemakers, Or cerebral aneurysm clips made of metal cannot be examined using MRI because the machine's magnet could potentially move these objects within the body, causing damage.
How do cells communicate with each other?
Cells communicate with each other via small signaling molecules that are produced by specific cells and received by target cells. This communication system operates on both a local and long-distance level. The signaling molecules can be proteins, fatty acid derivatives, or gases. Nitric oxide is an example of a gas that is part of a locally based signaling system and is able to signal for a human's blood pressure to be lowered. Hormones are long-distance signaling molecules that must be transported via the circulatory system from their production site to their target cells. Plant cells, because of their rigid cell walls, have cytoplasmic bridges called plasmidesmata that allow cell-to-cell communication. Animals use gap junctions to transfer material between adjacent cells.
What are linked genes?
Linked genes are genes that tend to be inherited together. The human genome has 24 chromosome types and approximately 35,000 genes, therefore, at least 1,000 genes would, on average, be found on a single chromosome and travel through meiosis as a discrete unit. However, linked genes tend to be close enough that they are separated by crossing over less often than unlinked genes.
Do all cells have a nucleus?
Prokaryotic cells do not have an organized nucleus. Most eukaryotic cells have a single organized nucleus. Red blood cells are the only mammalian cells that do not have a nucleus.
Who is considered the father of zoology?
Aristotle, 384 to 322 b. CE, is considered the father of zoology. His contributions to zoology include vast quantities of information about the variety, structure, and behavior of animals. The analysis of the parts of living organisms, and the beginnings of the science of taxonomy.
What is biomagnification?
Some compounds are not recycled by decomposers, nor are they released into the atmosphere like energy. Instead, they remain in the ecosystem in virtually unchanged form as they are passed from one organism to another by predation. If a larger fish consumes 5 smaller ones every day for several years, some of the compounds in the flesh of those little fishes will be transferred to the larger fish as it builds and repairs its own structures. Over time, the larger fish will accumulate many units of such compounds. An example of these compounds is the pesticide DDT. The toxic effects of DDT may not be apparent in the small concentrations found in the little fishes, but the accumulation over time in the larger fish will allow the effects to be magnified. This may become even more apparent as the chemicals move up the trophic pyramid to the top predators like the birds, or humans, that eat those larger fishes. To describe this phenomenon, ecologists use the terms biomagnification and bioaccumulation in recognition of the disproportional effect these toxins have on the upper levels of the ecological pyramid.
Do bears in a zoo hibernate?
Bears do not technically hibernate. In winter conditions they find protected areas like a cave or hollow log and conserve energy by taking extended naps, winter sleep. This is why it is always dangerous to disturb a wintering bear, because it is merely napping, not hibernating. In zoos, because temperatures in cages and enclosures remain warm throughout the year, and the bears have access to a food supply continually replenished by keepers, they remain active year-round.
What are the basic steps to transfer bacteria using aseptic procedures?
First, plug the test tube or flask with cotton or plastic cap. Sterilize the inoculating loop or needle, tool used to transfer the bacteria, by heating the wire in the flame of a Bunsen burner or alcohol lamp until red hot. Remove the cotton plugs from the test tubes or flasks, and briefly flame the mouths of the test tubes or flasks. Pick up the organisms to be transferred by dipping the inoculating loop or needle into the liquid culture or touching the growth lightly. Transfer the culture to a new test tube by dipping the loop gently into the broth or drawing it across the agar. Pass the mouths of the tubes through the flame again and then reinsert the cotton plugs to prevent the entrance of other microbes. Heat the inoculating loop or needle again to destroy any remaining organisms. The procedure to transfer a sample to a petri dish is similar except the culture is streaked along the agar in a pattern to isolate the bacteria on the petri dish.
What is a mermaid's purse?
Mermaids' purses are the protective cases in which the eggs of dogfish, skates, and rays are released into the environment. The rectangular purse is leathery and has long tendrils streaming from each corner. The tendrils anchor the case to seaweed or rocks, where the case is protected during the 6 to 9 months it takes for the embryos to hatch. Empty cases often wash up on beaches.
When was the microtome invented?
Although there were cutting engines as early as the 18th century. The first microtome was invented by Wilhelm His, 1831 to 1904, in 1870. Its development allowed scientists to prepare very thin, uniformly sized slices of tissue for examination under a microscope, instead of the imprecise ones previously prepared using a handheld razor or cutting engines.
Are there reliable rules to identify poisonous mushrooms?
There are no general, reliable rules to identify poisonous mushrooms. Some of the edible varieties are quite easily recognized. But some edible varieties closely resemble poisonous mushrooms and can only be distinguished by an expert. The common lore that poisonous mushrooms make silver spoons turn black, while mushrooms that can be peeled are edible, is not true. Some of the deadliest mushrooms, amanitas, do no turn silver spoons black and can be peeled. The only rule to follow is that one must be able to identify a mushroom with certainty prior to eating it.
What is delayed implantation?
Delayed implantation is a phenomenon that lengthens the gestation period of many mammals. The blastocyst remains dormant while its implantation in the uterine wall is postponed for a period of time lasting from a few weeks to several months. Many mammals, including bears, seals, weasels, badgers, bats, and some deer, use this phenomenon to extend their gestation period through delayed implantation so they give birth at the time of year that offers the best chance of survival for their young.
What are autosomes?
Autosomes are chromosomes that contain information, gene sequences, available to both sexes. These are the chromosomes commonly used in genetic examples.
What are some examples of genetic engineering in plants?
Genetically engineered plants include transgenic crop plants that are resistant to herbicides used in weed control. These transgenic crops carry genes for resistance to herbicides such that all plants in a field are killed with the exception of the modified plant. Transgenic soybeans, corn, cotton, canola, papaya, rice, and tomatoes are used by many farmers in the United States. Plants resistant to predatory insects have also been genetically engineered.
Why do enzymes only work in specific environments?
Because changes in temperature and pH can cause the structure of a protein to change. Every enzyme has criteria that must be met in order for it to perform its function. For example, the amylase that is active in the mouth cannot function in the acidic environment of the stomach. Pepsin, which breaks down proteins in the stomach, cannot function in the mouth.
What sources do muscle cells use for energy?
Muscle cells use a variety of energy sources to power their contractions. For quick energy, the cells utilize their stores of ATP and creatine phosphate, which is another phosphate-containing compound. These stored molecules are usually depleted within the first 20 seconds of activity. The cells then switch to other sources, most notably glycogen, a carbohydrate that is made of glucose molecules strung together in long branching chains.
What is a stone plant?
The species Lithops turbiniformis is known by several common names, including stone plant, flowering stone, and living stone. The genus name Lithops comes from the Greek words lithos, which means stone, and opsis, meaning face or appearance. The plant is native to South Africa, Namibia, and the west coast of Africa. The plants are very difficult to see since their appearance mimics the rocks and stones of their natural habitat. They have two thick leaves and a single flower that is either yellow or white in color. Stone plants thrive in very arid conditions with minimal rainfall.
What is hermaphroditism?
Hermaphroditic animals have both male and female reproductive systems. Hermaphroditism provides a means for animals to reproduce sexually without finding mates. For example, individuals in many species of tapeworms fertilize their own eggs. In other species such as earthworms, each individual serves as a male and female during mating, both donating and receiving sperm.
How was it discovered that microorganisms are effective against bacterial infections?
British microbiologist Alexander Fleming, 1881 to 1955. Happened upon the discovery of penicillin's use as an antibacterial agent. In 1928 Fleming was researching staphylococci at St. Mary's Hospital in London. As part of his investigation, he had spread staphylococci on several petri dishes before going on vacation. Upon his return he noticed a green-yellow mold contaminating one of the petri dishes. The staphylococci had failed to grow near the mold. He identified the mold as being of the species Penicillium notatum. Further investigation proved that staphylococci and other gram-positive organisms are killed by P. notatum. It was not until the 1940s that Howard Florey, 1898 to 1968, and Ernst Boris Chain, 1906 to 1979, rediscovered the benefits of penicillin and were able to isolate it for medical use. In 1945 Fleming, Florey, and Chain shared the Nobel Prize in Physiology or Medicine for their work on penicillin.
How were bacteria involved in World War I?
During World War I the British needed the organic solvents acetone and butanol. Butanol was required for the production of artificial rubber, and acetone was used as a solvent in the manufacture of the smokeless explosive powder cordite. Prior to 1914, acetone was made by the dry heating, pyrolysis, of wood. Between 80 and 100 tons of birch, beech, or maple wood were required to make 1 ton of acetone. When the war broke out, the demand for acetone quickly exceeded the existing world supply. However, by 1915 Chaim Weizmann, 1874 to 1952, had developed a fermentation process by which the anaerobic bacterium Clostridium acetobutylicum converted 100 tons of molasses or grain into 12 tons of acetone and 24 tons of butanol. British and Canadian breweries were converted into acetone and butanol factories until new fermentation facilities could be constructed. Commercial acetone and butanol were made by this fermentation process until they were replaced by cheaper petrochemicals in the late 1940s and 1950s.
What is bioremediation?
Bioremediation is the use of organisms to remove toxic materials from the environment. Bacteria, protists, and fungi are good at degrading complex molecules into waste products that are generally safe and recyclable. Sewage treatment plants perform bioremediation in a limited way. An example of bioremediation is the massive cleanup in Alaska following the Exxon Valdez oil spill in 1989. The superficial layer of oil was removed by suction and filtration. But the oil-soaked beach was cleaned by bacteria that could use oil as an energy source.
What are the problems an animal must overcome to move?
In contrast to other organisms, animals are able to move. The two forces an animal overcomes to move are gravity and friction. Aquatic animals do not have much difficulty overcoming gravity, since they are buoyant in water. However, because water is dense, the problem of resistance, friction, is greater for these animals. Many of them have sleek shapes to help them swim. Terrestrial animals tend to have fewer problems with friction. Since air poses fewer problems of resistance than does water. However, terrestrial animals must work harder to overcome gravity.
Who introduced the gypsy moth into the United States?
In 1869, Professor Leopold Trevellet. 1827 to 1895, brought gypsy moth egg masses from France to Medford, Massachusetts. His intention was to breed the gypsy moth with the silkworm to overcome a wilt disease of the silkworm. He placed the egg masses on a window ledge, and evidently the wind blew them away. About 10 years later, these caterpillars were numerous on trees in that vicinity. And in 20 years, trees in eastern Massachusetts were being defoliated. In 1911, a contaminated plant shipment from Holland also introduced the gypsy moth to Massachusetts. These pests have now spread to 25 states, especially in the northeastern United States. Scattered locations in Michigan and Oregon have also reported occurrences of gypsy moth infestations. The gypsy moth, Porthetria dispar, lays its eggs on the leaves of oaks, birches, maples, and other hardwood trees. When the yellow hairy caterpillars hatch from the eggs, they devour the leaves in such quantities that the tree becomes temporarily defoliated. Sometimes this causes the tree to die. The caterpillars grow from 0.5 in, 3 mm, to about 2 in, 5.1 cm. Before they spin a pupa, in which they will metamorphose into adult moths.
What are beta blockers and how do they affect cell function?
In order to understand how beta blockers work, one must understand that most drugs work by binding to receptors on the cell membrane. Drugs called agonists activate cell receptors by causing either an increase or decrease of cell function. Beta blockers are a class of drugs called antagonists. Because they block agonists from binding to cell receptors. An example of a beta blocker is propranolol, which is used to treat high blood pressure and angina. Propranolol works by protecting the heart against sudden surges of stress hormones like adrenaline.
What effect does aspirin have on enzymes?
Aspirin is probably the most common over-the-counter medication. Aspirin blocks the production of cyclooxygenase, COX, enzymes. COX-1 and COX-2 are two important enzymes with different functions. COX-1 catalyzes the biosynthesis of hormones important in maintaining the stomach lining. COX-2 catalyzes the biosynthesis of chemicals that promote inflammation, fever, and pain when an injury occurs. The positive effects of aspirin, pain and inflammation reduction, are due to the blocking of COX-2 enzymes. However, the negative aspects of aspirin, stomach problems, are due to the blocking of COX-1 enzymes.
How long can bacterial spores remain dormant?
In 1995, scientists revived bacillus spores from the digestive tract of an amber-encased stingless Dominican bee, Proplobia dominicana, which lived from 25 million to 40 million years ago. Previously documented records based on ampoules that Louis Pasteur collected indicated that spores were able to survive for only 70 years.
Can organisms with more than 2 sets of chromosomes reproduce?
Organisms with an odd number of chromosome sets cannot produce gametes with a balanced assortment of chromosomes. Polyploidy is very common in plants, with 30 to 70% of today's angiosperms thought to be polyploid. When a tetraploid, 4N, plant tries to breed with a diploid, 2N, plant, triploid offspring are formed. An example of this is the seedless grape.
Biome summary. What is a cline?
A cline refers to geographic variation due to a gradient of climatic features. For example, a north-south gradient may include a range of temperature and a range of plant sizes, with taller plants at the southern end and shorter plants at the northern end.
How can chromosomes become damaged?
Chromosomes can become damaged physically, in their appearance, and molecularly, in the specific DNA sequences they contain. Chromosomes can break randomly or due to exposure to ionizing radiation, which acts like a miniature cannonball, blasting the strands of DNA. Other factors such as physical trauma or chemical insult can also cause breaks. When a chromosome breaks, the broken part may then reattach to another chromosome, a process called translocation. Sometimes the broken chromosomes may attach to each other, forming a ring. Any type of chemical change is known as a mutation.
What is meant by common origin?
The term common origin refers to the shared ancestors of the Darwin-Wallace theory. It explains the commonality we see among different species of birds, For example, blue jays, starlings, sparrows, and other birds share traits like beaks, wings, and general body structure that are of common origin.
What is artificial selection?
Artificial selection is the selective breeding of organisms for a desired trait, such as breeding a rose plant to produce larger flowers or a chicken to lay more eggs. Darwin cited artificial selection as evidence that species are not immutable, that is, unable to be changed by selection.